Hi, in this video we'll talk about our third cytoskeletal filament, actin, both as an independent cytoskeletal element and with its very important interaction with myosin, which promotes muscle contraction. On this slide we see that actin filaments are preferentially located just inside the plasma membrane. They can form dense networks or somewhat linear bundles. They exist inside the microvilli on the border of epithelial cells and at leading edges of cells as they undergo motion on a flat surface. The filaments themselves are narrow. They're 7 nanometers. They're the smallest of the filaments. It is a repeating subunit of a single protein, the actin protein. Like microtubules, there's a plus end and a minus end, so every subunit is oriented in the same fashion. And we'll see that there are lots of proteins which actin can interact with, including myosin for contraction, but many other proteins which regulate its structures and its function. Here we see some of the different configurations of actin and function, as we mentioned at the microvilli of the epithelial cells, as contractile bundles which can cause cells to change shape. At protrusions of leading edges of direction, the cell is crawling in this direction. And during cytokinesis, during mitosis, at a contractile ring, it's actually the filament that is contracting, that is pinching off the cytoplasm between the two cells. Like microtubules, they are dynamic in that they form and dissociate, and they can be stabilized into structures by many of the binding proteins. Actin polymerization holds the SPE record for filament formation. When a sperm makes contact with an egg, from the acrosomal head of the sperm, it will open up and it will project an actin protrusion to make contact with the egg. This is 90 microns long in 10 seconds. That's a rate of 1.2 inches an hour. And that represents the incorporation of about 3,000 monomer units per second into a growing filament. Like microtubules, whose polymerization rates are modified by the binding of GTP and the hydrolysis of GTP, actin uses ATP as a nucleotide factor which helps stabilize interactions. So here's a plus end and here's a minus end. This is the same subunit repeated over and over. The dark red color is where it's still ATP, and then as it hydrolyzes, it loses an organic phosphate. In contrast to microtubules, which only grow or shrink at the plus end, actin can grow or shrink at both ends. Subunits can come on here as ATP, and they favor sticking here, but they can also dissociate. Actin with ADP bound can also associate with a growing actin filament, but the equilibrium favors dissociation. So the strengths of interaction are different depending on the state of the nucleotide. This slide also depicts the slightly helical nature of the actin filaments. As they extend, they will rotate around, sort of like in a rope-like twist. This is another slide. It does show an electron micrograph, but other than that, there's not a great deal of new information here. Even though these subunits are different colors, they're all the same protein. And in this case, the plus end is down here at the bottom. Actin is capable of interacting with a great many number of proteins that will influence its function and structure. The amount of actin monomers influences the rates of polymerization by simple mass action, and so that the amount of free monomer is regulated. It's held at a much smaller percentage than microtubules, which was about 50% monomer. In order to regulate the amount of monomers, actin can bind to sequestering proteins which are sort of taken out of circulation and then don't contribute to the equilibrium between the polymerized state and the monomeric state. There are proteins which cause actin filaments to associate. They can associate in bundles and parallel strips. They can be cross-linked to form gel-like structures. There can be capping proteins which will stabilize the ends and allow them to persist for long times. There are severing proteins which cleave actin, which can take a gel-like structure with a lot of cross-linking and quickly reduce its velocity. There are proteins that promote the growth of actin filaments. And finally, most well known is that it interacts with motor proteins. In this case, myosin is pictured to form contractile bundles. On this slide, we see a depiction of a cell moving thanks to actin protrusion. You might think of the analogy here of a person inside a large plastic bubble, which moves by walking on the inside. You'll get the ball to roll. It's a little more complicated here. First of all, cells do stick to the surface, and we'll learn more about this in the last video for this section, is their interactions with the extracellular matrix. This substratum, perhaps in a laboratory experiment, would be some form of collagen. And so there's a sticking to the surface so that the cell is not allowed to slide freely on the surface. But what happens is you get actin polymerization on the leading edge. This will cause this edge to protrude and go forward. It will form another anchor attachment, and this will sort of solidify the extension that has occurred. Meanwhile, there'll be contraction on the leading edge, and this is actin contracting here with interacting with myosin, and then the interactions here with the extracellular matrix can be severed or broken, and then it will rotate forward until it hits the next attachment point, and then the cell will continue to protrude again, and the cycle can repeat itself. Here we see an electron micrograph of the dense gel-like network that can occur at the leading edge of a crawling cell. And we can see here that the most active polymerization is occurring right here at the edge, and that these blue capping proteins will help stabilize the positive ends of actin, and so that as they degrade, they're degrading primarily from the minus end. So as the cell moves forward, you continue to have a layer of highly polymerized gel-like actin just beneath the surface. On this slide, we see two interesting images of the exact same cell, which really allows one to visualize how the actin network interacts with these extracellular matrix anchorages to help the cell both maintain its position and then move forward when it wants to move. On this picture here, we see sites of attachment. These are the anchoring points between the cell and the surface. On this site, we see actin bundles, and here we see the actin filaments that are originating from this anchorage point, and here we see the ones that originate from this anchorage point. And this makes sense structurally, that the dynamic moving elements are connected to the solidifying or structural elements, allowing for the most efficient locomotion for the cell. As is probably well known to most of you, myosin is the motor protein that associates with actin. You'll remember that microtubules had kinesins and dyneins for motion in the positive and negative direction along microtubules. Here we see the structure of a monomer. It has a long tail region. This tail region is used to allow myosin monomers to interact with each other. There is the hinge region. This is the area where you have a conformational change, which will cause the head to move positions. And then this is the head of the motor domain. This is the part that interacts with the actin filaments, the part that does the walking. Here's different ways that actin and myosin can associate. Myosin can be connected to the plasma membrane, and actin can be pushed parallel here. It can also be used to move vesicles, but with myosin walking along the actin filaments, though you should really think of the kinesins and dyneins walking along microtubules as the major interaction for most of vesicle motion inside a cell. Like the kinesins and dyneins, there are many different types of myosin that are used in special situations, but we'll talk on mostly the basic one, which is myosin 1. Actin and myosin are organized into myofibrils, or sometimes called muscle fibers. These are actually large multinucleate cells and they're arranged in sarcomere subunits. Here, we'll see this later, but these are the Z discs. These are the sort of anchorage points of the muscle. Here you have actin fibers attached to the Z discs, and then in between here you have the myosin, and these probably actually go, the actin probably goes a little further, and here you have myosin overacting, interacting with these actins, and these set of myosins are going to walk this way along the actin filament. These myosins will walk this way, and that has the effect of pulling these two Z discs closer together, since this distance is fixed between the two sets of heads. And here we see the myosin fiber. We see many different myosin filaments, and we can see them interacting. Again, here's the tail units, the hinge unit, and the heads. Here's the midpoint. All the myosins on this side have the heads here and the tails like this, and on this side you have tails and heads here, and in the so-called bare region they're just overlapping tails. And here we can see how contraction can occur. In this case we have unfixed actin filaments, so they will be sliding past each other. This actin filament is interacting with this head. It's being pulled this way as the myosin crawls out here. This myosin is crawling this way, pushing the actin filament this way. And if these ends were fixed, then this would have the effect of pulling this end closer to this end. But in this case, they're just sliding past each other. And here we see some details of the actin-myosin interactions in a sarcomere unit in sort of higher resolution. That you can see that when contraction occurs, that these myosin heads have walked many times up the actin filament, and now there's a greater overlap. You can see right away how come you can have stronger muscular contraction, not when the arm is fully extended, but when it has already started to flex a little bit, and now it can generate more force because there are more heads interacting with the actin filament. And since the shape of the myosin subunits, the length of it cannot change, as they walk this way and that way, they're actually pulling the Z discs closer together. And we saw in the previous electron micrograph that there are many of these sarcomere units next to each other, so it's the simultaneous contraction of all these sarcomeres that produces the muscle contraction. A few interesting points about this process is that each step for each of these myosin heads, as it moves or walks one step along the actin filament, that hydrolyzes an ATP, so you can see that there's going to be lots of ATP being hydrolyzed very quickly. Think too about how fast you can flex your arm, and that has to in order to do that, these myosin heads need to make many simultaneous steps. Each step of myosin is about 5 nanometers in this direction, or 50 angstroms. And the movements of the myosin heads themselves are unsynchronized, so that there's always a large number that are attached while others detach and are walking and taking a step, so that you can maintain a state of contraction and not lose it if all the myosin heads would dissociate at the same time. Each myosin filament has about 300 heads interacting with actin, and each head can contract every 200 milliseconds, or about 5 times per second. The sarcomere units are typically about 3 microns when fully extended, and about 2 microns when fully contracted. I think when you contemplate the number of interactions going on between myosin and actin, the fact that each step requires an ATP hydrolysis, how many heads are interacting just even in a single sarcomere, and then how many sarcomeres are involved in a muscle fibril inside a muscle, and that each step requires the hydrolysis of an ATP, it sort of boggles the mind at the amount of ATP production that is required to power even a single muscular contraction. On the final slide, we'll talk a little bit about the molecular mechanism of muscular contraction, or the interaction between myosin and actin, and as you've seen, actin is sort of a passive filament here, and it's the myosin that's doing all the work. Here again, the hinge region being the place where these structural changes are occurring. And we talk about how the cycle of this power stroke, if you will, requires ATP, and sort of the surprising thing about the molecular details is the power stroke occurs while the myosin is bound to ADP. That triggers it. It does the power stroke. It's going to release the ADP, and the myosin head will stay attached to the actin filament as long as it is not nucleotide bound. When ATP comes in, it is the molecule which will cause the release of the myosin head from the actin molecule. The myosin will hydrolyze the ATP, allowing the myosin head to relax or extend, and as it loses phosphate, the inorganic phosphate, then just bound with ATP, it will reattach. It will do the power stroke as it's releasing it, and this cycle will repeat over and over again. So without ATP, you are locked in this fully contracted state. This is the cause of rigor mortis upon death. And while it is perhaps somewhat surprising that the ATP step is not powering the contraction or the power stroke, remember that this is a cycle, that biochemical reactions can be coupled, and unfavorable reactions can be powered by favorable ones. The most energetically favorable is the hydrolysis of the ATP. In each of these steps, you have a slightly different configuration or electrostatic environment. Losing the inorganic phosphate, that changes the amount of negative charges here at the motor head and is partly responsible for the contraction step. The loss of ADP, you're losing a lot of interactions with the myosin protein, and as well as a lot of charge when ADP dissociates. So again, I think we can rationalize this all as changes in protein conformation based on simple thermodynamic and electrostatic considerations. Anyway, that concludes most of what we want to say about the actin filament. Thank you for listening.